here at the University of Guelph at the Biodiversity Institute of Ontario. The goal is to catalog all the biodiversity on the planet. It's exciting because of what it means to the world.
I think that DNA barcoding is such an amazing new technology. I've been working at Bio since 2006, and just to see how everything, where it started and where it is now, is just amazing.
DNA barcoding is a method to identify species.
In order to accomplish that, we help people collect data and we do development in order to get more insights into these big data that people have gathered all around the world.
What I do is I array all the specimens into 96 well microplates, and there's a ton of insects in each of our samples. And DNA barcoding is extremely awesome because I cannot tell myself what everything is in that sample. DNA barcoding, we just all put it in the plates and it does the trick for us. It'll tell us what that species is exactly.
What I really love about DNA barcoding is such a cool tool for helping to identify species way faster than we have in the past.
The downstream goal is to actually have a handheld device so that scientists, researchers, field workers go into the field, out in the field, and basically analyze a sample on the spot to know more information about the sample that they've recovered.
The most amazing things were always when you discover something new, a species you would not have thought of has been not encountered before. And a lot of times it's actually, for example, fishes that people have been seeing for years and years. They all look very much alike, and then it turns out with the help of DNA that a fish that looks always the same is actually 2 or 3 more species.
I think what we differ from other people is that we not only provide the lab, the collection team, and also the informatics support, we also provide them insights about the data because we care about the data, so we actually have teams that are focusing on helping the user going from the beginning of their research all the way to the end of publication. So I think that's really the main big difference between us and other people.
It's building that reference library. So having a lot of DNA sequences for everybody available on the planet through an online database.
The Biodiversity Institute stands out among other facilities around the world because we're a high throughput facility. We deal with tons of specimens.
We have gotten through a lot of samples that are in upwards of 100 years old and have been able to process and get great results. That's the most exciting thing to me is coming from a background of high throughput genomics is that we're constantly pushing the envelope for new technologies.
I don't think there's any other facilities around the world that are as fast and high throughput as ourselves.
What we're doing has some relevance to society. It's more than just my own curiosity. It is something that will help society control things better, avoid certain things. So it really makes a difference.
Hello everyone, I hope you enjoyed that video introduction to the Biodiversity Institute of Ontario. You're tuned into today's PIR live event and I'm your host, Scott Jones. Remember that you can tweet your questions anytime during the presentation today using hashtag AskPIR, and I'll read them to our guests during the question and answer period afterwards. If there's room in your tweet, please include your name, school, or city so we can give you a shout out. Our guest today then is Dr. Dirk Steinke. He's a senior researcher at the Biodiversity Institute of Ontario and the Centre for Biodiversity Genomics at the University of Guelph. I look forward to getting to some of the questions on Twitter, but first I'll turn it over to Dr. Steinke so he can tell us a bit more about his research, including some citizen science work.
Yeah, thank you very much, Scott. Because I don't have much time, it probably best is I show you a couple of slides I prepared for today. They should help you to understand a little bit of what we're doing. The video before showed you already what our institute is probably well known for. It's a method called DNA barcoding that helps us to identify species. And you can do a lot of things with that. And what I'm trying to do is give you one example of what we're using DNA barcoding for. We've been doing that for now, 12 years or so. And that particular method has also something to do with schools, because we decided we need to get some help with what we're doing. So what I'm trying to tell you a little bit about is something that has to do with a very common thing that happens throughout the country and in other countries as well. We call that environmental assessment. So anytime when humans or when people in a country or in a region decide to protect a certain area of land because they want to keep it intact and pristine, so keep it in its original state, or when you do the opposite, if you're trying to use a piece of land, for example, if you have a piece of forest and it's supposed to be used for logging, or you want an area and extend your agriculture, or you want to build houses, urban development, or if people want to extract resources like mining, anytime they're trying to do these kinds of things, there's a couple of laws that require them to make what we call an environmental assessment. That means that experts like us have to go to these places and find out what lives there. So we have to determine the biodiversity of that region that's supposed to be reused for something else. And we have to make a judgment of if that is actually possible. Because imagine that there's probably a couple of species living there that live nowhere else in the country or nowhere else on the planet. Then it's probably not such a good idea to use that land, and probably move to another region where this is not the case. And all these questions require an expert advice. And some of these methods, like logging or mining, require the companies also, once they're done and finish their job, to restore the area, really go back and try to bring it back to its original state, although they have been working there for years. And also that needs to be assessed and how successful that is. Normally, for the most part, people have been doing that by using indicator species. So those are species that define characteristics of the environment, and you pick a few that are easy to find or easy to identify, and then you make a judgment of, yeah, if they occur here, this is probably a very good sign. There's also a couple of indicator species. When you pick those, they're rather a sign of a very disturbed or harmed environment. But it's usually just a few species compared to all the ones that are living there, and there lies the problem. If you have just one or two species of an area, you might make the call that everything is fine, I've seen them, but you don't know a lot about the rest that lives there. Quite honestly, most of these species we overlook from doing that are very tiny, hard to identify when you just sit down and try to do that by eye, even for experts for these particular groups. So that's why this DNA barcoding you heard about before, so the use of DNA sequences to identify species probably becomes in handy. And the dream we have and the things we're developing for is being able to do these kinds of assessments and give the people who needed the answer, these are all species that live there, an entire list of them. And the question is, well, one question we probably answered already, we use the DNA barcoding method to do that. And another thing I like to talk about is how do we get all these species? It's probably easy to observe these biggies, but what about a lot of small insects? That's where this trap comes into play. So this is one of many traps we're using here in our institute, and a lot of researchers around the world do the same thing, and that's called the Malaise Trap. It has nothing to do with the French word malaise, that means sickness. It's actually in honor of the guy who developed it, a Swedish person called René Malaise. So the idea is that you put that somewhere where you think insects fly back and forth and around, and then this mesh in the middle of this tent-like thing is sort of an obstacle for insects. And most of them, by instinct, would try to overcome that obstacle by flying towards the sunlight. And there's a white mesh on top that intensifies the signal. And now imagine a fly comes along, flies there, encounters that, and the only way out of this thing is into this little bottle up there. And that's filled with a preservative liquid, which is good for our DNA work, which is in most of the cases ethanol. So now we have a lot of these flies and insects trapped in there. Sometimes the predator will chase them there as well. So sometimes you find spiders and similar things. And just to give you an idea how good these traps are, just look at these jars. So this is what we call an insect soup. And each one of those jars is about a week of catch with one of those traps. So you can see you catch a whole lot of them, sometimes thousands of individuals in a single week. And that makes us very confident that we cover a lot of these specific groups of animals in one go. And imagine you have that out for a season, summer in Canada, May to September, October. So what we usually do. So you have a lot of these bottles to go through to determine what lives there, what originally lives there. And then later on, if people come back with their assessment, they might say, hey, look, things have changed over time because something happened, some sort of pollution, some other environmental impact. So we can make these determinations now just using a relatively easy to maintain trap map. So we started doing that here, then we switched over and thought, well, we can do that Canada-wide to establish a baseline of what lives in Canada. And you see a lot of photos here of Malaise traps, and we actually covered pretty much all the Canadian national parks over a 3-year period to collect as much as we can and identify them and put them in our online database. And you heard about that one in the video. But we thought we can do more. We need more help. It's just a handful or probably a couple of dozens of researchers that can do that in Canada, and we have partners across the world. But how about including guys like you? So in 2013, we started what we call the School Malaise Trap program. And what we do is, for 2 weeks, we give schools one of those traps, and they collect insects the same way we do it, which means they have these bottles at the trap, and they change it after 1 week, so that at the end of 2 weeks, they pack the trap and the 2 bottles that are full of insects, hopefully, and send them back to us. And what we do is sort everything that's in there, select particular insects, do the DNA extraction, and read the DNA information we need for our barcoding work, and then we send the school back a long, long list of specimens. You see a lot of numbers here in that slide. The most important ones are probably the ones, how many species we actually were able to find with that. So we were doing that for 2.5 years, roughly, almost 3. Over that time, we collected and barcoded about 60,000 individual insects, and that's equivalent to 6,500 species. The fascinating thing and the good thing about that is, and that shows you that this is very important for us as well as scientists, having these data from schools and our school students helping us, is that about 1,100 of those species were species we were not able to find before, so they were new in our database. Most of them, we probably knew they might occur, but we never encountered them. Others were new to Canada. Others are new to science even. So there were a couple of schools that found species that had been never encountered before. So it's a pretty cool program in many respects, and even for us, because all these data go into the same database as the ones that is contributed by our scientists. And we open our registration, by the way, today for our fall run of the program. So we will do the same thing in September, October of this year. So if your school is interested, our website is open and the registration as well. Just to give you an idea of what the findings were, before I quickly wrap up to allow you a lot of questions, is over the course of the project we've been doing that, the Canadian terrestrial arthropod species list said Canada has about 32,045 arthropods. And arthropods means, in that case, insects, spiders and their relatives, millipedes, centipedes, crustaceans, and all that. So when we started doing our national park program and the ones with the schools, we already found almost 38,000 different things, meaning that the species count is a little bit, no, not a little bit, a lot underestimated. Canada has far more animal species than we thought it has. And again, the trap itself captures only a fraction of them, only the ones that fly around and happen to end up in this trap, which is a lot. It's probably the best trap that's out there to collect these kinds of life forms, but it covers only a portion of the cover. And the last thing I'd like to show you is we didn't stop there. We didn't say it's enough to involve schools in there. We also thought, hey, we would probably give it to kids and their parents or families when they go out there. So we have this live scanner kit, which is essentially 4 vials, where somebody can put samples in there. And by the way, people not only use it to find insects or identify them, they go to a restaurant, they want to know if their fish fillet they just ordered is really the species that was written on the menu, because sometimes it's not true. And then they can send us these samples, and all the data they collect go through an iPhone app, are sent to us before. And then we can do our usual laboratory routine and then send back the result, the identification, what it was, back to the cell phone the information came from. So that's my little talk. I'm finishing out here, sharing the slides. 1 sec. and then I'm back.
Okay, great. Thanks, Dr. Steinke. So just a reminder to everyone listening that you can tweet your questions using the hashtag AskPIR at any time for the remainder of our half an hour here. In the meantime, I'm wondering what sort of applications do you see for such a database of DNA barcodes?
Well, I showed one, but there's far more. One of the things I just touched on, and it has been used extensively for food safety. So very oftentimes we find that producers or supermarkets often have food items that could be plants, any kind of veggies or fruits, or it could be meat. And they put a name, what species it's supposed to be. Is it beef or is it that type of fish? And then sometimes you find out it's actually not this one. And there's a bit of fraud going on. And very often you find, for example, fish is substituted for each other. And usually you have a very expensive fish you buy or you order in the restaurant. And what you've been served is a much cheaper version of it. And it's hard to identify if you have a fillet in front of you and it has been fried or whatever. How can you tell what it was before? And the same goes for a lot of different products. So there's a bit of safety in that. And there's a couple more forensic applications. Even criminal forensics is looking into that because they'd like to know certain insect types to determine time of death or certain environments and to confirm that. There's a lot of different things. Honestly, we didn't think about them initially. We thought it's a very good way to actually figure out how many species live on Earth because that's an unresolved question. But then over time we figured, oh, you can do this with that and you can do all these different applications.
Okay. So we do have a question here, it looks like from Madame TB's grade 5 class in Waterloo. And they want to know, what is the average number of insects that you are able to trap per day in your tents?
That can vary very much on what kind of schoolyard, for example, it is, on what kind of environment we are. We had actually schools where we caught over 10,000 individuals per week. So you can do the math divided by 7 and you'll get the daily volume. So it's over 1,000. But it can also be that you trap only, let's say, 40, 50 a day. We'd have 2 runs per year. There's one in spring and one in fall. Usually we catch far more in the fall because our spring run runs very shortly after the winter stops. So in some of the regions in Canada where we had traps, the schools actually put them out when there was snow. So the daily volume was probably 5 insects. But in other regions, it was dozens of them.
Okay. So once you do have these insects, I presume you then take them back to the lab to do the DNA barcoding. How long does that process take to do the actual barcoding?
Usually I would say on average, the process sorting, pulling a piece of tissue, in the case of an insect, that's one of its legs or a piece of it, and then going through the laboratory routine, it can take up to a week. If we have customers, with paying customers, I'd say, industry, for example, and they're really in a rush, we could do that in a day. That's possible. And there are newer machines out there that promise, but they haven't been really delivering yet, to do that within a few hours.
Okay. I know you have tents at schools around Ontario. What is the difference you see from tent to tent? Like, is it very region specific in terms of the insects that you'll end up receiving?
Oh yeah. So we have it not only in Ontario but Canada-wide, but there's certain differences. It's not like every school, even in close proximity, let's say Greater Toronto region, would have the same species in there. There's always some sort of variation, especially a variation between a city and a rural region where the school is out there with just a very small township or whatever around. So there's a big difference there. They share some, but there's also great differences between the amount of species, but also which species are in there. Okay. It's not that you could say, hey, there are more species, it's more diverse somewhere in a rural forestry region than, for example, in the middle of Toronto. We've seen both very diverse areas in the middle of this big city. The only thing that's different is what species you find there.
Okay. I know you mentioned in your talk that some of the insects you haven't even seen before and nobody has discovered before. I'm sure that might surprise some of the people listening today. How is that possible? Is it just that our technology is catching up to the biodiversity right now, or?
Yeah, likely. The technology is certainly catching up. It allows us to do what we've been doing for over 200 years much faster than we used to do it. And we put more effort into really collecting systematically. In the past, you had a lot of researchers, they have their pet group, particular group of animals or plants or fungi they're interested in, and that's what they collect. But what we do is trying to get broad and collect pretty much everything we can get with certain methods. So we're catching up in that front, but we're still not the awesome power force out there, because currently people are trying to estimate how many species are there. We have, within about 250 years, discovered and described 1.8 million species. But the lowest estimates of species on Earth are about 7 million, 7.5 million, 10 million, depending on who you're talking to, that can go up to 100 million. And 2 weeks ago, some researchers did an estimate of how many microbes, so bacteria alone, they think we find on Earth, and they came up with a staggering number of 1 trillion just species of microbes.
Wow.
Which is, even with our modern technology, an impossible task. But if we focus on everything that's not on bacteria, I guess we can catch up a bit faster than the 250 years that got us to this point.
Okay. So yeah, that's obviously a very staggering number. Does it become, what about storing all of this data? Is it computationally possible to store all this data at the moment in one place, or?
No, well, the amount of data we've collected, and we're talking about right now a little bit over 5 million records, 5 million barcodes for about 520,000 species. That's here in our building. Downstairs is a server array, and we have mirror sites and 2 different others, places in China, one in Netherlands, just to make sure that the data is not lost. But with all the modern works of cloud computing and everything else, storing the information won't be the problem. But right now, the new technology that allows us to do DNA sequencing at a much higher rate, so you can generate millions of them in one single run. There we get to the limits that the computer actually can help us sorting them out and sorting them into the right bins. So what happens is that you have a certain waiting period until you get your results, which requires patience, but it's not impossible.
Okay, good. What about, I'm sure you trap a bunch of other things besides insects. Do you trap and barcodes non-insect species then?
Oh, yeah.
Okay.
I think I mentioned it. Sometimes you get the odd spider ending up in the same bottle, which is kind of fascinating. But what happens is that the spider likely followed its prey and insect in there and ended up in the same trap. We had one school last year, which was kind of funny. They found 2 praying mantises sitting right next to the entrance to the bottle. And they were picking up the insects that were flying into our trap, just getting a few here and there. I think the students were really mean. They just grabbed the 2 praying mantises and threw them in the bottle before they shipped it to us. But these were insects as well. What we find is very tiny guys that you wouldn't expect to be in there, like colembala, or that are known as springtails. They're usually soil-living little creatures. They crawl all the way up to the Malaise Trap. We're talking about an animal that's probably about 1 millimeter less size, making its way up this trap, which is probably about a 1-person height. So you can imagine, if you translate that into human dimensions, you would be having somebody climbing up all the way to Mount Everest to jump into this bottle of preservation liquid. So those are mostly the things we cover. Of course, other animals, other groups of animals, we have different methods. For everything that, for example, doesn't fly, we usually use pitfall traps. Because they will never end up in a Malaise trap. So when we go out collecting for arthropods in general, we have an array of trapping methods that catch as many as we can.
Okay. Can you tell us maybe a bit more about your group at the university and also at the institute? With that much data that you're collecting, I'm sure it's an enormous task to be able to sort through it and make some analysis of it.
Yeah, well, about us, currently we're about 120 people here in this institute.
Okay.
And that's a very diverse group of people. You have us researchers that sort of steer the boat and figure out the best ways of collecting and what to do, and doing the statistical analysis to figure out have we done enough or not. Then you have a lot of actually programmers and database architects and all that, so the modern IT jobs, they take care of the database. And that's about 20-something people that have to make sure that this database keeps running. We have 15,000 users, I believe, in that database worldwide. So we're not alone, we're not the only ones, but we're running most of these systems. And then there's a lot of people that do exactly what would happen when somebody participates in the school Malaise Trap program. They receive the bottles, they sort through them, and they prepare them for the lab, and there's another group of people taking over and doing all that. So we can, or we have been doing 1 million of these barcodes in the laboratory per year for the last 2 years. So that's our equipment. We have a lot of robotics. You've seen that in the video, perhaps, some of the robots moving liquids. That helps a lot. So that's what this group is mainly doing. But then, of course, with so much data and so much information, we can do a lot more things in terms of science. So we're trying to figure out things we didn't know before, as usual for a scientist.
Okay.
And they not necessarily have to do with biodiversity assessment or so.
Okay.
With the others.
One of the things, so it's obviously really neat what you're doing with the Citizen Science Initiative. Do you ever foresee, once you have a large enough database, making use of citizen science to help with maybe some of the classification of the insects? Is that somewhere you could see this going?
It could, actually, and sometimes it really does. Our problem is the amount of experts for particular groups, especially in insects and other smaller things, is relatively limited. So we're talking about the job of a taxonomist, and I think we have about 10,000 on Earth, and most of them are older than 55. So that means there is an entire group of people who know what species they're looking at by comparing their experience in that, and we're losing them. But on the other hand, side of things, a lot of the taxonomy we have, and I talked about these all described species and discovered ones, a lot of the insects, for example the beetles, have been discovered by amateurs, by scientifically interested citizen scientists, but not necessarily by one professor going out in the woods and trying to find them. No, it's people who like that kind of work and really study everything, and they have been helping all along. So I'd say a good chunk of the 1.8 billion species we know so far has been because of that. And the modern methods don't stop them from doing them. They can still help us putting a name on whatever DNA barcode we have generated.
Okay. Looks like we have another question on Twitter here. How is the process of DNA barcoding different than, say, mapping a genome, which we hear about often?
Mapping a genome means you really are interested in obtaining the entire DNA information of an entire genome. DNA barcoding is picking a tiny fragment in comparison to that. Just to put that into context, the human genome is about 3 billion base pairs long. So you have these letters, 3 billion letter book, how to construct and how to maintain and how to run a human. The barcode region we use is about 650 of those 3 billion. So a very tiny fraction, which makes it also easier to retrieve it. And it's really just for that purpose. There's other purposes for other gene regions or other DNA regions. For example, if you talk criminal forensics, you want to put a person to a place. So for example, my barcode region I'm working with cannot distinguish between one and another human. I can only tell the sequence I have in front of me is from a human or from a chimpanzee or an elephant or a fly. But there's other DNA regions people could pick, and then they can say, yeah, this was this human, this was this human, and they can place people to a crime scene or to a certain area. So we're really just picking a small fraction, not a very large genome.
I see. Okay, so we're almost out of time here. I'll give you one more question I like to ask all of our guests, which is, for the students listening, what is the subject areas that they should focus on if they want a career in DNA barcoding?
If they want to go there, there are several avenues. I think the most safe ones is either you do environmental science or you go the biology route. So very often people from our zoology or integrated biology department end up having jobs here. And as I said, environmental science people as well. So that's usually what you have to do. You go to university, you look for your Bachelor of Science program. It might be an integrated biology one. It could be an environmental science one, or genetics and genomics very often provide a similar route towards us because we need experts on all ends. And as you heard, even somebody with a computational informatics background can end up here, because that's what we need when we run these large databases.
Okay, perfect, perfect. So unfortunately, that's all the time we have today. So I'll just do a quick plug for our next event, which is Thursday, June 2nd at 1 p.m., when we'll discuss antibiotic resistance with Dr. Kimberly Harcombe from MacEwan University. In the meantime, I'd like to say thank you to all of our live stream viewers, and of course to you, Dr. Steinke, for taking the time to share your expertise and answer our questions today. Thank you very much.
Thank you. Thank you for having me.
All right, bye for now, everyone.